Tonight's agenda, in our previous live, we did DNA and RNA structure and function. We did DNA replication. So we also did transcription. What's on the docket for tonight is translation and the genetic code, and then a really cool topic, regulation of gene expression in operons. If we go further, we'll get into eukaryotic gene expression, but I think we'll probably just do items 4 and 5. I'm doing DNA and gene expression because I did a poll on YouTube a couple of days ago, and there was an overwhelming desire to do this topic. And after this, I think, again, what I'm hoping happens is that as you get more into the review that you're doing in your classes, wherever you are around the country, I am hoping that you'll just come with questions, and then it'll be me responding to questions, and that will be an even better way to do it than the way we're doing it right now. So just to introduce myself, I'm Glenn Wokerfeld, also known as Mr. Dev. Recently retired from teaching at Berkeley High School. When I taught at Berkeley High School, most of my kids got 4s and 5s on the AP Bio exam. That is for many reasons, but one of them is that I used the learn-biology.com website, which I wrote. Why I wrote it for my students, and then I used it with my students. And you can sign up at learn-biology.com/apbiology. I've already told you all the great things that are in there and how it's going to promote your learning biology. Your subscription to learn-biology.com comes with a subscription to the Biomania AP Bio App, which puts many of the resources of learn-biology.com in your pocket. So I hope you take me up on this offer, and I hope you purchase the subscription. I know that some of you who are viewing have, and the more that do, the better, obviously, it goes for me, but the better it'll go for you too. It's really all about you and your journey towards success in AP Bio. One thing that I want to encourage you to do, if you haven't done it already, is I have an AP Bio checklist, and you should download it because it's a great way for you to self-assess what you're doing. I have another video that I recently published. It's an AP Bio plan. And in that, I have an entire plan for you to study for the next 2 weeks, or for the 2 weeks preceding the AP Bio exam, which means starting tomorrow. That's when the plan begins. So tomorrow you would be on day 1 of the plan, and you would use the checklist to do a self-assessment. So I hope you do that. The reason why I have this screen grab from the College Board's personal exam description is just to let you know that I'm not making any of this stuff up. We're doing it because it's in the curriculum. And the stuff that we're going to do tonight actually has an extraordinarily high likelihood of being in some form on the AP Bio exam. So that's the story. We're going to start with the genetic code and translation / protein synthesis. And here are the questions we'll be addressing in this topic. What's the genetic code? How does it work? What are the key molecules and structures involved in protein synthesis? And describe the process of protein synthesis. So the genetic code is represented in a variety of ways. The one that's actually become most common is this circular representation of the genetic code. And the genetic code is the code used by all living things to translate nucleotide sequences into amino acid sequences. And again, what we need to be doing at this point in the year as we approach the AP Bio exam is making cross-topic connections. So an important piece of evidence for the evolutionary idea that all life is created is the idea that all living things use the same created genetic code. In other words, if you think about terms from evolution, this is a homologous feature shared by all living things. When the first cell crawled out of a hydrothermal vent 3.8 billion years ago, that cell used this genetic code over here. So how does it work? Basically, there are groups of 3 codons. Like, for example, AUG, that's a codon. Why? Because it codes for one amino acid. This is a word that was formulated when this process was figured out. AUG codes for one amino acid. Which one does it code for? Well, in this genetic code, here's the first amino acid. The second ring out here is the second amino acid. And the third ring out is the, um, I've been using the wrong word. First nucleotide, second nucleotide, third nucleotide. So AUG, therefore it's methionine, all right? So in other words, what's AUG code for? Methionine. What is GUC code for? I'm gonna go to the first nucleotide, the second nucleotide, and the third nucleotide. GUC is valine. So using that method, throw this out. Will you have to do this on the AP Bio exam? I think there's a high probability of that. And I don't like making predictions about what will be on the test and what won't be on the test. This is such a foundational skill that I think you can count on this one being on the test. Um, so things to know about this code is that it's nearly universal. There's a couple of microorganisms that have some slight variations on this code that you see here, but it's pretty much shared by all life. It's specific. In other words, if you have these letters, if you have A and G, you know exactly what amino acid it codes for. But even though it's specific and unambiguous, it's also redundant. So there are synonyms. And what do I mean by that? Well, GUG spells out leucine, this nucleotide, but so does GUA, also codes out leucine. And in general, the third nucleotide in a codon is the one that's most forgiving. And when we talk about mutations, we'll see there's a kind of mutation called a silent mutation, which has no impact on the amino acid. It's usually a change in the third nucleotide in a codon that causes that. See if there's anything else to say. So here's the solution to this problem. AUG, GUC, AAG, GUU, those RNA nucleotides code for methionine, valine, lysine, and just to be clear, spend no time memorizing the genetic code. No, 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 no. You will always be given the genetic code. I think it's more likely that you'll be given a rectangular code, a tabular code, as opposed to one like that. I don't know if I do. Fantastic. So here, let's use the genetic code dictionary to code out CUC. So how do you use this one? The first nucleotide base is over here. Here's the second base, and here's the third base. So if you want to code out CUC, you go CU, and third base over here, that codes out to leucine. All right. GAU, got a G over here. I got an A over here. And now I'm looking for my U, G, A, U. No. GAU. That intersects over here. Asparagine. Fun fact, asparagine, asparagus. I mean, this is now going to be studied on the internet, but this is the amino acid that street after eating asparagus. It smells like asparagus. So, fun fact, now you know, asparagine smells like asparagus. And so on. So GCA, you use the same method. So CUC is leucine, GAU is asparagine, GCA, G, C, and now I'm looking for my A. GCA is, let's see, G, C, A. I'm looking up here, and that is, did I get that right? GCA is alanine. Okay. All right. So anybody have any questions about this? You know, how you use the genetic code? It's a foundational AP biology skill. You got to know this. Okay. And here's the whole answer. All right. So that's the genetic code. That's really what you need to know about it. And I'm not seeing any questions, and I'm assuming that that means that we are all good. So the big picture of translation. The big idea is that mRNA is synthesized in the nucleus. It goes out to the cytoplasm. Of course, I'm talking about prokaryotes, where it's met by a ribosome. These codons, such as this at N, they specify the order of amino acids. The ribosome connects amino acids to create a polypeptide. In other words, in the same way as in DNA replication, the DNA replication, DNA polymerase didn't know which the next nucleotide is. The information was in the molecule that's being replicated. The ribosome doesn't really know which is the next um, tRNA that's going to come in carrying the next amino acid. That information is specified by the mRNA. There's a variety of tRNA molecules that are floating around. The only ones that will bind are the ones that have a matching anti-codon. So tRNAs, shown at letter O over here, they bring amino acids, like here's one shown over here, to the ribosome RNA complex. And they have an anti-codon binding site. And again, if you can sort of imagine this, many tRNAs try and bind with CCC. Only ones that will stick will be the ones with GGG. Why will they stick? This is again a foundational idea of biology because of complementary shape and hydrogen bonding. Okay, so that's the basic idea. Um, so what is the role of the ribosome in translation? The ribosome is a general purpose protein factory. I think of it as a little nanoscale robot. And basically what it does is it uses the information in this tape, which is mRNA, to determine which amino acids are going to be strung together in a polypeptide. You can think of them as enzymes that string together amino acids to form polypeptides. They have a large, small subunit and 3 tRNA binding sites. And I like the acronym EPA, like the Environmental Protection Agency, to remember that. That might be useful to you, it might not be. E for exit site, P is for the peptide site, A is for amino seal site. We'll talk about how that works in a minute. And you don't really need to know that. You just need to know this at a slightly more superficial level, and I'm about to demonstrate it. So E is the exit site, P holds the polypeptide, and A gets the new amino acid. So how does translation begin? An mRNA will come out of the nucleus, and a small unit of the ribosome will bind with the mRNA at the start codon. So the start codon is almost always AUG. So you can sort of imagine the ribosome small unit sliding along the mRNA until it finds AUG. You think of it like an enzyme, well, that's where its active site is, and that's where it stops. I've reached AUG. Okay. Then the small unit sits there, and then what happens is that a tRNA carrying the amino acid methionine and with the matching codons. This is AUG. You have to think AUG. A is going to bond with the U. U is going to bond with A. G is going to bond with C. So we've got this anticodon UAC that comes in and binds. And so now we're almost done. But what's going to happen is the large subunit of the ribosome binds with the small subunit. And note that this first tRNA is in the P site, E, P. So we've initiated translation. We don't have a protein yet. All that we have is one amino acid that's sitting there. How is the process going to continue? The process continues with elongation. And isn't that wonderful? Initiation, that's how it starts. Elongation, that's how the protein gets longer. It completely makes sense in terms of the terminology, unlike other things in biology. Like you just have to use brute force to remember like the gold complex and things like that. But it all makes sense. So what happens next is this site was open, right? This site was occupied. A tRNA comes into the A site bearing the new amino acid. So based on which one binds? Well, the one with the anticodon that matches this codon. So this next codon is GUC. It's going to be matched by CAG. And I'm realizing you probably can't see those letters on your screen. Maybe you can, but just know that it's an anticodon that matches this codon over here. So now we've got these two amino acids that are adjacent to one another, but they're not bonded. And that's where the ribosome carries out its function. The ribosome acts as an enzyme that catalyzes a peptide bond between the first amino acid and the second one. So now we have methionine bonding with the valine. Ribosome catalyzes the peptide bond between the P and A side amino acids. And then I didn't really represent this well, but I'm going to show you a little animation of it in a minute, I think. Um, the ribosome translocates. In other words, everything moves over. So here's AUG, here's UUC, and now we've moved over to the next codon over here. So now we have AAG, and everything's shifted over one side. So this was over here, now it's over here, this is over, and now the A side is empty. All right, so we've started the elongation process, and now we've got to continue it. Well, this tRNA that was in the E site exits. A new tRNA enters at the A site. So here's a new tRNA coming in. So here's the exit. Here's the new tRNA coming in. And the ribosome then catalyzes another bond. So it catalyzes this bond between the second amino acid and the third one that was carried in, lysine. Why lysine? Because lysine was carried in by a tRNA that has a matching anticodon. And that, by the way, is specific. Like there's an anticodon amino acid match, and it's not really part of the AP curriculum, but there's actually another amino acid that can basically, excuse me, another enzyme, it's a protein enzyme that can essentially know, oh, this is the tRNA, this is the amino acid that I've got to put on over here. And all these enzymes are just doing this by shape and by feel. So the process continues, and we're going to make our protein longer and longer. So there's just continued rounds of translocation, exit, and then the next tRNA with the next amino acid. That can go on for hundreds or thousands of amino acids because proteins are big. And they can be composed of thousands, even sometimes, I believe, tens of thousands of amino acids, but certainly hundreds. This is happening at an enormous speed, and that's how these ribosomes are working with mRNA and tRNAs to churn out proteins. And like again, you ever think like, oh, these silly pictures, it doesn't matter to me. It's not important. This is happening. It's happening in you right now. Right now, all, if not most of your cells are carrying out this process, building proteins. All right, so any questions about initiation or elongation? Okay, let's go on. Finally, we get to the termination phase of translation. So initiation, elongation, termination. Termination, another beautiful way, easy to remember, to terminate things is to end. When your employment is terminated, you lose your job. Termination is the end. I'm trying to think of other words. So terminal. Well, terminal is like the end of the line, kind of. So describe the termination phase. At a certain point, the ribosome reaches what's called a stop codon. So the genetic code that you saw a couple of slides ago, it's got codes for amino acids, and it also has punctuation. And when I say punctuation, I mean like a period at the end of a sentence. It's not a word, but it designates that the sentence is over. So the ribosome reaches a stop codon. There's no corresponding tRNA. And instead, what comes in is a release factor. I drew this. I've been working with these diagrams for a long time ago, and then Harry Potter came along, and then the movies came along, and all of a sudden, my students were telling me that this looks like Gandalf's hat. It wasn't really intentional. I didn't mean it to look like Gandalf's hat, but I guess it does. like Gandalf's hat. That's the release factor. And of course, that's just an arbitrary shape. It's a protein. It occupies this last um, codon over here, the stop codon. And essentially what it does is it says, folks, the game's over. We're done with protein synthesis, and let's now end the process. So what does ending the process mean? Well, first of all, this polypeptide, this string of amino acids, is released into the cytoplasm where it's going to fold up. And the truth is that it starts to fold up right away. And if you remember, I did an earlier show about protein folding, a video about that too. But what does that mean? That the secondary and tertiary interactions start to happen, so that this linear thing folds up to its three-dimensional shape. And of course, another thing related to biology, structure determines function. So that's how the structure of the protein emerges, and that's how it does its particular job. What might be the job? Proteins are receptors. Proteins are enzymes. Proteins form structures like your fingernail and hair, which I don't have a lot of, but like the lens of your eye. Pretty much everything in an animal is protein. A tiny bit of carbohydrate, stuff like that. So the release factor comes in, the ribosome dissociates, the mRNA goes into the cytoplasm. Sometimes that mRNA is destroyed by enzymes. Sometimes it might be actually read again by another ribosome. But basically, release factor enters, ribosome dissociates, polypeptide, and the polypeptide folds into the functional protein. So I've got a song about this, and I'm going to try and play it. And maybe this is going to be very interesting. I'm going to need your help because things, as you know, go very well to start this session. I'm really hoping that you're going to hear the sound.
Look at what's coming out of the nucleus, destined for a ribosomal strand of mRNA. It's got the code, the information for the protein we'll be making today.
And I'm worried that sound is
the RNA message is organized into codes. That's a sequence
of
bases 3 that spell out one and only one amino acid, a genetic code that works universally. A codon has its match in an anticodon, 3 bases on tRNA's bottom side. tRNA's job is to bring amino acids to ribosomes so proteins can be synthesized. mRNA, tRNA, and ribosomes plus amino acids make a protein-making machine. Read the codon, make a polypeptide, fast translation, cell synthesized protein.
That was what I wanted you to watch. I wanted you to see that animation because that's what's happening. And that's how you can see how the ribosome is translocating. New tRNAs are coming in carrying new amino acids. And that's how the entire process unfolds. So this is my protein synthesis song. And if you want to learn more about protein synthesis, that is a great way to do it.
The small subunit of a ribosome.
All right. So any questions about protein synthesis? Maybe I'll jump back a slide or two. Like any questions about any of those phases? Just put those questions in the chat and nothing will make me happier. And again, I hope I wasn't tense at the start of the show or anything like that, but like nothing makes me happier than when I hear your questions or read your questions in this context. So anything? Okay, so we're going to go on and we're going to talk about operons. Yeah, thank you, Sophie. The release factor is, paradoxically, it's a protein. And so the release factor is floating around in the cytoplasm at some concentration, so where it's high enough so that when the ribosome reaches the end of the mRNA and this release factor is in the A site over here, there's no tRNA that can bind over here. No tRNA has a matching anticodon to UAA. But the release factor has a matching shape. Of course, it doesn't have an anticodon, but this is a shape. It's some kind of molecular shape. And this protein has a molecular shape where one part of it matches with it. So it's kind of a little bit mind-bending and it feels a little circular, but the release factor is also a synthesized protein that's made through protein synthesis. So that's the basic idea. That's where the release factor comes from. And in like all of these tRNAs are waiting. They're just sort of randomly moving around in the cytoplasm with their amino acids. And again, the incredible thing about biology is that there's like all of this randomness inside the cell. You have all of these tRNAs, they're bound to amino acids, they're moving around randomly, but the mRNA imposes order process. So the only tRNAs that can bind are the ones with the matching anticodon, and that's true for the release factor too. It binds because it has a shape that matches the stop codon. So Sophie, I hope that answers your question. That's where it comes from. That was a great question, and every question will be a great question because they're so fun. So any other questions about protein synthesis? Okay, fantastic, Sophie. You're welcome. Okay, so here's the deal. It's 7:48, and usually at this time I'd be moving it to multiple choice questions, but I hope people can stay a little bit longer because I started this like 20 minutes late because of my technical snafu at the beginning of this show. So I hope people can stay. I want to talk about operons, and then we'll do some multiple choice questions. We'll end at about 8:20, and I hope that works. I know that if any of you are on the East Coast, then that's probably getting a little bit late. I mean, you're teenagers. You don't go to sleep at 7 o'clock or 8 o'clock. You probably go to sleep at like 12:30 or something, and really not so healthy like that. Do people want to put in the chat what time they go to sleep? That would just be an interesting thing. So, I go to sleep really early. I'm in bed by like 10:20, something like that. Okay, we did this. We're not going to do this movie now. No. Okay, operon. I love operons. I have always, the entire time that I've been studying biology, I have thought that operons are the coolest thing, and they are. So in this topic, talking about operons, what are the questions that we'll be addressing? What are operons? What's the difference between an inducible and a repressible operon? Some of you here got a little bit of that last night, so it'll be a little bit of review. Those are the folks who are participating in my Zoom reviews, which again, they're open to everybody. Oh, wow, you guys stay up really late. Okay, all right. And Berkeley High, I hope you don't have the zero period at Berkeley High School because that was just brutal. Berkeley High, for those folks who are not Berkeley High people here, they have a zero period of AP Bio that starts at like 7:20 in the morning. I think it's like a bad dream. I barely remember it anymore. But anyhow, that's not really good for teenagers' bodies. But that's really not what we're talking about today. So, how does the trip operon work, and how does the lac operon work? And so that's the cool stuff that we're going to do. So here's just a little bit of context. E. coli is the experimental organism that this stuff was discovered in, but it's a pretty typical bacterium. It lives in our guts. It lives in the guts of almost all mammals. It might even live in the guts of all vertebrates. But here's E. coli, and its chromosome consists of about 4,000 genes and 4 million bases, which, just for some context, it's only 1/1000th the size of our genome, which is 4 billion bases approximately. So 4,000 genes. They can't be expressed all the time. What's the system for turning them on and off? That's really what this lesson is all about. And gene regulation, super, super important topic in AP biology. And operons are like that. And Lily, I see that you've got a zero period, and I'm really sorry. But at least I know that I'm not going to be keeping you up really late because we'll be done by 8:20. So what's an operon? It's a cluster of genes transcribed as a single RNA. So in other words, here you have a bunch of structural genes, one gene followed by another, followed by another, followed by another. And those are all transcribed as one RNA transcript, which later gets processed into multiple RNAs, which produces multiple proteins. Okay, so that's one definition for an operon, a cluster of genes transcribed as a single RNA. But the more important thing to know is that it's a prokaryotic system of gene regulation that has control elements. So it solves the problem that I just mentioned. E. coli, 4,000 genes. Which ones get expressed and when? So here's the basic structure of an operon. There are structural genes that code for protein or proteins. There is an operator where a repressor protein binds. There's a promoter where RNA polymerase binds. And this like open DNA over here, that just means that many, many bases away, there's a regulatory gene. All right, and that regulatory gene, it might be 1,000 base pairs away in terms of the DNA. And that regulatory gene codes for a regulatory protein. That regulatory protein is the repressor. So, but things are a tiny bit more complicated, and we do a little bit of simplification in the AP Bio curriculum. It's a regulatory protein. It's usually a repressor, but regulators could also stimulate. So that's what we got. So we're going to start with the trip operon because its operation is a little bit simpler than the lac operon. So here's basically how the system works. It codes for a series of enzymes that make tryptophan. What do I mean? That doesn't even sound like English. Tryptophan is one of the 20 amino acids, and bacteria need it to make proteins. Bacteria can synthesize tryptophan from simpler precursor molecules when there's no tryptophan freely available in the environment. So again, one thing that you have to sort of be thinking about is that everything I'm talking about happens inside a bacterial cell. So this is gene regulation that's happening inside a bacterial cell. So that means this is inside a bacterial cell. This is the DNA of the bacterial cell. So it codes for a series of enzymes that make tryptophan and acid. And if tryptophan is absent, if there's no tryptophan in the environment, then there's a regulatory protein. Here it is over here. It's made by the regulatory gene. It can't bind with the operator. The operator is a series of DNA nucleotides. That means it has a specific kind of shape, and proteins can bind with that shape. There are many proteins that bind with DNA, but the default form of this repressor is can't bind. Okay? What that means is that RNA polymerase will bind to the promoter, and it'll just go down the length of the rest of the operon, and it will transcribe the structural genes. And that's what we see happening over here. So this is what happens when there's no tryptophan in the environment, and E. coli needs tryptophan. But what happens when there is tryptophan in the environment? And again, here's how you got to think about this. This E. coli can be inside you, right? It lives in the colons of mammals. You're a mammal. E. coli, in fact, lives in your gut. You have E. coli living in your gut now. If you were to eat a turkey sandwich, turkey is protein, it's muscle tissue. It's relatively high in tryptophan. Then this would be a E. coli situation. It would be in your gut, surrounded by tryptophan, and that tryptophan would be diffusing into the cell because things diffuse from high concentration to low concentration. And again, if this was Zoom, we could do this really quickly, but it's a little bit awkward. But like the question that I would ask right now is just looking at tryptophan, when tryptophan diffuses into E. coli, is that facilitated diffusion or simple diffusion? I'm going to take a sip of tea from my Science Music Videos mug while you answer that question. Is it facilitated diffusion or simple diffusion? Fantastic. Way to go. And it is facilitated. And the reason why you know that is you see this hydroxyl group over here and this carbonyl group over here. And you might not even know what their names are, but you got to know like a little bit about functional groups. I mean, this amine, all of this stuff makes this part of the molecule polar. And polar stuff does not diffuse through the lipid bilayer. It needs to be facilitated. So again, this point of the course, we got to connect everything. So now we're connecting what's happening with operons to what happens with cell membranes. So all of this tryptophan is diffusing in. So that means that inside the cell, there's going to be a lot of tryptophan. That tryptophan is going to bind with the repressor protein, and it's a protein. When something binds on one side, it changes shape on the other side. And suddenly, that can bind at the operator. And now what happens? Bam. Tryptophan binds with the repressor protein. The repressor changes shape. It binds with the operator, and that blocks RNA polymerase. It's such a beautiful system. If tryptophan is present, the cell should not make it. It should not synthesize tryptophan. Okay, so that's the rule. This is about cellular efficiency. This is an adaptation that promotes survival. So TRP is a repressible operon. Why is that? Because the presence of tryptophan turns the thing off. It represses the system. Repression. Repression is when you like curtail people's freedom, when you have a repressive government. So repression turns things off, keeps people from doing things. Repression, stopping. Okay. So tryptophan acts as I think it's actually called a co-repressor because it works with this repressor molecule to turn the system off. Good, got that right. Okay, complicated stuff. Let me just pause for a second. Anybody have any questions about how the TRP operon works? All right, let's look at the lac operon and then we'll do some multiple choice questions. So the lac operon is for a different thing. The lac operon is for making enzymes. It's an inducible system, and it's for making enzymes that digest lactose, a disaccharide. Lactose is a disaccharide, but disaccharides can't be fed into the cell for glycolysis. For glycolysis, you need monosaccharides. So basically, you got to break this bond over here, and there are enzymes that can do that. So what happens when lactose is in the environment? Again, what does that mean? That means that it's in your gut and you take a drink of milk. Milk, the sugar in milk is lactose. And this, by the way, has nothing to do with lactose tolerance or intolerance or something like that. This is about E. coli and what it digests, not what you can digest. So when lactose is in the environment, it diffuses into E. coli. And then what will happen is lactose will bind with the repressor protein, and that will keep the repressor protein from binding with the operator. That enables RNA polymerase to transcribe the genes for lactose digesting enzymes. And that is so cool because when lactose is absent, there's no lactose to bind with the repressor, and the repressor binds with the operator, and we can't transcribe enzymes to digest lactose. That makes so much sense evolutionarily. Why produce enzymes to digest something when it's not there? But the repressor's default shape lets it bind with the operator in that case, and RNA polymerase can't transcribe the structural genes. So this has a different rule. If lactose is absent, don't make the genes to digest it. And that makes lac an inducible operon because lactose acts as the inducer. These are all negative feedback systems. Again, connecting what we're learning in operons to what we learned in Unit 4 about feedback and homeostasis. Why is that? Because what the system does is it turns itself off. So if there's lactose in the environment, what happens? We produce enzymes to digest all the lactose, and then the system will turn off. Same thing with TRP. TRP's also a negative feedback. Why? Because when tryptophan is in the environment, it keeps the cell from making tryptophan. It turns expression of the system off. Same thing. Absence of tryptophan starts transcription. Transcription results in production of tryptophan, which turns the system off. Negative feedback. I'm going to skip this for right now because I know that 2 of you got it, but I can't resist playing just a little bit of my Operons song. This is a rap song that I released this year, and I just want you to listen to 2 minutes of it.
Hey, I'm E. coli, a bacterial cell. Gonna tell you about something I do so well. It's the way I turn my genes off and on. It's a regulatory system called an operon. My chromosome has over 4 million bases and 4,000 genes, so I need some bases for deciding which gene gets activated. And operons are how my genes are regulated. One operon has initials TRP, the trip operon. Its function, you see, is coding for enzymes that make tryptophan, an amino acid. Here's my plan. If there's tryptophan around, then E. coli shouldn't make it, so I shut the system down. But if tryptophan is lacking and it's something I require, I turn the system on and make what I desire. But how does the bacterium ascertain what and when to synthesize when it's got no brain? Let's examine the structure to get the information about the workings of operonic gene regulation. Operons have prokaryotes, turn genes off and on. A cluster of genes transcribed as a single RNA. Operons have prokaryotes, turn genes off and on. Repressing, inducing the operonic way.
Okay, enough of that. What we're going to do right now is listen to this. You're going to love this. Listen.
AP Bio Review.
Yeah, AP Bio Review.
Choice questions.
Okay, so I am going to go to learn-biology.com / I'm just going to go to learn-biology.com. Okay, so good. I'm clicking on the AP Bio Review button. And, again, want to emphasize that everybody can have this experience without me. Like, you don't need me to do these multiple choice questions. To do these multiple choice questions, you just need to go and say to your mom and dad, hey, it's $24.95. We spend money like that all the time. And it would really, really be helpful to me if I could do these multiple choice questions or these flashcards or anything else. So we're going to do some multiple choice questions right now. And here we are, bam, multiple choice review. And probably not logged in. Okay, I'm in. All right, so okay, stop that right now. All right, so we've got a question picker. So what you do is you go like this, expand gene expression. And what did we do tonight? We just did, I can't limit it to operons, unfortunately. So we might get some other gene regulation questions. We'll deal with it. This is an operon question. Fantastic. How lucky. Okay, let me make this a little bit bigger. Okay, so everybody lean in, take good luck, and let's read this. The diagram below shows the lac operon in the presence and absence of lactose. The genes LAC-Z, LAC-Y, and LAC-A allow the bacterium to use lactose as an energy source. Which of the following accurately connects the structure and function of the repressor molecule? 1. When the bacterium needs energy, ATP binds the repressor. The RNA polymerase to transcribe the genes to break down lactose and make ATP. 2. The repressor molecule has 2 binding sites, each with a correct shape and charge to bind either DNA or lactose. When lactose is present and binds the repressor changes shape so that it can no longer bind DNA, allowing transcription to take place. The repressor molecule, I noticed the applause, Sophie and Lily, thank you very much. The repressor molecule has a site with the correct shape and charge for binding DNA and a second site with the correct shape and charge to bind lactose. When lactose is not present and is not bound to the repressor changes shape so that it binds DNA, allowing transcription to take place. And then 4, when the bacterium doesn't need energy, lactose changes shape and charge so that it binds to the operator. Since the operator is bound by lactose, the repressor binds to the RNA polymerase, preventing it from attaching to the DNA. As a result, transcription does not occur. Folks, uno, dos, tres, or cuatro, which is the answer to this question? All right, we've got two. I've got two choices for two. Anybody else want to weigh in? Let's click on two. Excellent. The function of the lac operon is about the repressor having binding sites for lactose and DNA. When lactose binds with the repressor, it causes an allosteric shift that prevents the repressor from binding with the operator. Let's see that. When lactose binds with the repressor, causes an allosteric shift that prevents the repressor from binding with the operator, allowing RNA polymerase to transcribe the gene for lactose digestion. Okay, let's do one more. Okay, the nucleotide sequence below is part of the template strand for the gene coding protein Q. The G on the left is at the 5' end, and the A is on the right, the 3' end. The first amino acid is coded from GTT. What is the second amino acid that this DNA is coding for? You can use the genetic code below to answer. This is a slight tricky question, so just be careful as you code things out. What is the second amino acid that this DNA is coding for? I'm just going to give you a hint. This requires two steps. And if you're like me, you're going to have to write it down a little bit. I'm writing it down. Okay, so here's how you got to approach this question. This is DNA. It's not RNA. And the way you know that is because of the presence of these T's. So CGT is going to code for GCA. Okay? GCA. So now let's code that out. GCA. Alanine. And alanine is the other answer. And you've got to really be aware, like, totally, this could be an AP question. Many of my questions are modeled after AP questions. So alanine is indeed the answer. I just want to show you, like, what would have happened if you got it wrong, though. Like, if you did put GLN, glutamine. You get a hint. The second amino acid will be the one coded from the second triplet, CGT. Find the mRNA complement of CGT and then look up that code in the genetic code chart. So again, this is how I wrote Learn-Biology. Wrong answers get hints, and those hints will guide you to the correct answer. Okay? One more question, and we're going to shut things down for the night. Oh, fantastic. We are in luck. It's another Operon question. Which of the following is true of the repressor molecule? All right. Is it an amino acid, a small RNA, binds to the operator region it's coded for by a structural gene? So, uno, dos, tres, or cuatro. 1, 2, 3, or 4. Let's see the answer. Okay, I'm seeing threes. And indeed it is. Okay, fantastic. So I want to jump back to the slideshow. And let's just make a couple of closing moves. So the first closing move is that if you haven't already done it, download the checklist. The checklist, if you've seen my movie, The 2-Week Plan for Getting Ready for the AP Bio exam, the first step is to do a self-assessment with the checklist. So go to apbiosuccess.com/checklist, and the checklist guides you to improve the gaps. In other words, you'll find the gaps in your knowledge, and then you can start to fill them. And keep attending these live reviews. There's a schedule at learn-biology.com/live reviews. And tomorrow night, if you're a paid subscriber, we're going to do a Zoom meeting. And otherwise, we'll see everybody else on Thursday night. And what I'd really like to have happen is that if you're watching and you're not a paid subscriber, just like walk into the living room, find your parents, and just say, hey, $24.95 for this fantastic website and for access to Mr. W on Zoom. It's really going to help me improve my AP score. It might jump it from a 3 to a 4, from 4 to a 5. So I really want to encourage you, like, don't wait. Go over, do it now, bother your parents, get their credit card, and just make it happen. And then what you're going to do is you're going to go to learn-biology.com/AP Biology. And Sophie, I'm very sorry that you won't be able to be at the Zoom meeting. I'm hoping to see you at the next one. And I'm hoping that many of the people who are here will be able to make it, because again, $24.95, it's the best bargain in biology education. It's going to improve your score. It's going to improve your confidence walking into the exam. It's going to make it into a great experience. So we're going to end now. I want to thank all of you for, first of all, being patient with me at the beginning of this stream, which was, oh my goodness, such a disaster. But stuck with it. I hope that this was useful. Tomorrow night, we'll learn about eukaryotic gene regulation, and we'll probably get even through biotechnology, horizontal gene transfer. It's fantastic, fantastic stuff. It makes me love BIOLOGY. I am so thankful to you for being with me tonight. Please tell your friends to join these streams. And the next one of these is Thursday night. And, again, bother your parents, tell them, learn-biology, go to learn-biology.com/biology, and please sign up. So I'm looking forward to seeing everybody at the next one of these meetings. I'm hoping everyone who is here tonight will show up at the Zoom meeting. I know, Sophie, that you can't do it, but, we'll have others. And you can also, maybe I'll record it. But anyhow, everybody, be well. Good night. Everybody be safe. See you at the next session. Everybody take it easy.